Welcome back. Today we are going to discuss about the culturing of different types of epithelial cells. And you know that in our body there are different types of cells like muscle cells of the heart, which is highly specialized with branches, then epithelial cells, fibroblasts, nerve cells, etc. And you know that each and every cell has certain specialities. Their functions are different. Depending upon the functions, the nature of the cells are also different. And also the cells need a specific environment to grow. So based upon this, when we culture these different types of cells, the nature of culturing medium as well as the culturing surface also varies. So we will see one by one the necessity of different types of cultures. Today we will be seeing about the culturing of epithelial cells. There are various layers of cells which are formed by epithelial cells. They coat the surface of the body or the exterior of an organism, or the internal organs like nephrons, digestive tract, etc. And different types of cells are there, and the functions of, even though they are epithelium, their functions and their requirements and their nature vary. Sometimes they act as total barrier, such as the epidermis. When they are covering the body, it acts as a barrier of microorganisms as well as external materials into the body or from the body to the outside. So that acts as a barrier. So epidermis with a minimum permeation of polar substances or are regulated barriers. And for example, in the intestine and lungs, the intestine, only certain materials are absorbed with the different types of mechanisms, and the lungs also the exchange of gases. Carbon dioxide is given out as well as oxygen is taken in. So there is a differentiation, a nature of absorption or the nature of the barrier properties of these epithelial cells may differ. Then, in most of the cases, or in almost all the cases of epithelial cells, there is a polarization. So this picture you might be remembering. This is the picture taken from the kidney tubule. So kidney tubule is lined by epithelial cells. They have a basal side, that is the side facing away from the lumen of the tube, and the apical side or brush side which is facing towards the lumen of the tube. And these two sides differ in their properties. So there is a polarity, apical to basal polarity, and there is a basolateral side. So there is a polarity between the different sides of these epithelial cells, basolateral polarity. And epithelial, as I said before, epithelial has different functions. So they are associated with different functions like hepatocytes. They are involved in the liver metabolism. Then epidermal keratocytes. They have barrier proteins they have barrier properties. Then pancreatic acinar cells. They help in the secretion of digestive enzymes. Kidney tubular cells. We have seen it in the last semester. They help in the reabsorption of materials. So their functions are varied. So depending upon the functional variation, even though they are epithelial cells, their necessity as well as their physiological nature vary. So there are so many research activities are happening using epithelial cells. The focus of interest in the development of in vitro models for many years. The epithelial cells are used as in vitro models for studying different types of cellular interactions, cellular physiology. There are so many in the cancer cells. So because certain properties of these epithelial cells make them useful in these studies, like they are renewable. So they have the ability to proliferate. We can make a number of cells for your research. And they have certain specific compartments or stem cell precursors for the development of newer and newer or more and more epithelial cells. and they are very good models for studying the regulation of cell proliferation and differentiation. As I said before, they will be originating from stem cell niches in different parts of the body. So from the stem cells to the epithelial cells, differentiation properties, and also some of these epithelial cells can be differentiated into other types of cells using different differentiation mediums or substances. And their regenerative nature makes epithelial cell sites for malignant transformation. So many of the cancers are developed from the transformation of epithelial cells, like carcinomas of lung, breast, colon, prostate and bladder. That is another problem because they have self-proliferating ability and they can attain malignant nature. They can be transformed within the body into a malignant cell. So there, as I said before, they are used as appropriate models for in vitro studies like carcinogenesis. As I said before, many of the cancers are formed from the epithelial cell, the malignant transformation of epithelial cells, and they can also be used for the study of differentiation. This has produced many excellent models for differentiation. So that's why they are extensively cultured. And this provides some of the clear examples of stem cell maturation. Stem cell differentiation and maturation. These are clear examples. So in that way also epithelial cells are extensively used for research. Then the nature of epithelial cells. As I said before, depending upon the nature of the cells, the culture medium as well as the culture surface and also other chemicals used for the culture also vary. So we will see the nature of epithelial cells. So these are two epithelial cells. As I said before, they have a apical and basolateral polarity, and also they are connected by tight junctions. So there is a narrow inner cellular space. As a result, they are intimately connected or attached through these desmosomes and also the cytoskeletal fibers, protein fibers. So that is another important property of epithelial cells we need to take into consideration while we are culturing them. So they are intimately associated, and also we need to see that they are sitting on, or there is a basal membrane attached to the epithelial layer. So the basal membrane is formed by different types of proteins: collagen, laminin, fibronectin and proteoglycans. And many scientists have studied or they looked in the possibility of reconstituting this basal membrane so they can form a layer of epithelial cells outside the body and can be utilized for various purposes. Studying different types of research as well as also used to make like a sort of repairing the body. This can be cultured outside the body and can be fitted onto the body for repairing certain damaged part of the body. So this was tried. And the basal membrane is usually formed by the tight product of epithelium and the underlying stroma. As I said before, within the body they are closely associated. So that makes or that gives them a particular property that when we are growing them, they grow in sheaths. It is quite difficult to get unicellular separation. Separating the cells from each other is quite difficult in the case of epithelial cells because they are closely associated in vivo. And the regulated permeability and transportation, we have seen that depending upon where they are seen or what their functions, the nature of the epithelial cells differs, whether they can transport certain materials or allow the passage of certain materials or not. And as I said before, these epithelial cells are joined by desmosomes. These are the mechanical junctions connected to the intermediate filament cytoskeleton. So these desmosomes are connected to the filamental cytoskeleton. So that makes an intact or strong attachment between the cells. So this will hold the epithelium together. And this is a characteristic marker of epithelial cells. Because when we are growing, we need to see, sometimes different types of cells look in the same way. So we need to see whether they are epithelial cells or different types of cells. So this is a marker. If you can locate this intermediate filamental skeleton attached to the desmosomes or the presence of tight junctions with the desmosomes and the cytoskeletal filaments, that is a characteristic marker for epithelial cells. So this is already said. Certain areas epithelial cells acts as a barrier, like kidney, ductal epithelium or secretory acini. At these areas, the desmosomes provide very tight junction. It's a tight junctional complex, and it is specific to that particular epithelium. So between the epithelium also we can differentiate based upon the different types of desmosomes they have. So that is another peculiarity of epithelial cells. So the presence of these junctional complexes along with cytokeratin intermediate filaments, it provides us a very useful and specific marker for recognizing epithelial cells. And there are different types of keratins or different types of these cytoskeletal proteins and different markers that can act as different markers for different types of epithelial cells. That we will see in that detail later. Then, so culturing epithelial cells. So we have seen the cells are closely attached together, like through the tight junction, and they are sitting on a basement membrane, and sometimes they act as a they provide the free transport of materials, they help in the free transport of materials, or they have act as a limiting barrier. So based upon this, we have to adapt the culturing conditions. So they are closely associated in vivo and are strongly self-adherent. So that makes or that necessitates different mechanisms for their detachment and also for attachment. And they tend to survive better in vitro as clusters or sheets of cells because they have close intimate attachment to each other. So they can be grown as clusters or a sheet. As I said before, because of this close attachment or tight attachment, dissociation into a single cell suspension is difficult. So we need to take that into account when we are culturing the cells. And for the dissociation, we use gentle disaggregation mechanisms or collagenase. Normally in cell culture, we use trypsin for dissociating the cells. Trypsin is a bit harsh enzyme compared to collagenase. Here in the culturing of epithelial cells, normally we use collagenase. In certain cases, we can use, especially when dealing with cancer cells, we can use trypsin also. So collagenase and the physical disaggregation. Then we apply collagenase, and collagenase helps in the better survival of the cells compared to trypsin. That is why we prefer collagenase for the culture of epithelial cells, but their action is quite slow. So when you need a faster separation, you may use trypsin by adjusting the time of exposure. So you will get a mixture most of the time when you are isolating cells from a tissue. You get a mixture of cells. It may have both fibroblasts as well as the epithelial cells. So we need to separate them. So there are different methods of separation. We will see those different separation methods one by one. So here we have a suspension of fibroblasts as well as epithelial cells. There are one or two cells together or multiple cells together. So when we are suspending them in the medium, growth medium, what can happen? The epithelial cells are multiple cells together, so they will set even faster. So you just remove the supernatant and use the sedimented cells. That is one way. Or you can filter using gauze, allowing them to sediment through the more finely dispersed fibroblastic stromal cells, or by a filtration. And the fibroblasts, so when we are using the previous method, by allowing them to settle under gravity, still you will get fibroblast contamination. And you need to see the fact that the fibroblasts multiply faster than epithelial cells. So after a number of divisions, what happens, even though the initial fibroblast number is quite low, after a number of divisions, the number of fibroblasts increases because their multiplication rate is faster. They multiplicate faster than epithelial cells. So that will be a problem. So physical method of separation, that is the one method we have seen, sedimentation under gravity, is alone is not sufficient, because even if a small number of contamination is there, because of the more rapid growth rate, the fibroblasts will outnumber the epithelial cells. So they tend to overgrow the culture. So that will not help us. So we will finally, after a few days or weeks, you will get a culture with more fibroblasts than epithelial cells. And we can adjust the medium, like platelet-derived growth factors stimulate fibroblast proliferation. So you exclude that. As a result, fibroblast growth will not be supported. And also TGF, transforming growth factor, has cytostatic effect on fibroblasts. So you add TGF into the medium so that the fibroblast division will be suppressed. At the same time, your epithelial cells can divide faster. So by designing the medium, you can suppress the division of fibroblasts. In that way, you can reduce the number of fibroblastic contamination. And selective attachment. So you have a mixture of fibroblasts and epithelial cells. You seed the mixture into a flask and let the cells attach for 30 minutes or 1 hour. And what happens is, the fibroblasts attach faster than epithelial cells. So after 30 minutes, you take the supernatant out without disturbing the attached cells. So most of the fibroblasts are now attached to the flask. Then you repeat this process for the next 24 or 48 hours. After 1 or 2 hours or 30 minutes, you take the supernatant and move into the fresh flask. So the fibroblasts will remain attached to the flask. And as it comes towards the later stages, you will get more epithelial cells and less fibroblastic cells. That is one method of removing fibroblasts from the epithelial cells mixture. And the speed of attachment can vary. You need to see that also. If you have clumps of epithelial cells, so they settle quite faster, even faster than the fibroblasts, and attach to the flask. So you need to see the nature of the cell suspension you have got. The size of the epithelial aggregate, if it is bigger in size, it will favor faster attachment. And the physiological condition, the extracellular matter, how much time you have exposed the cell to the collagenase or trypsin. If you have exposed these cells for a longer period of time, the attachment will take longer, because most of the or a greater part of the extracellular materials are digested by proteins are digested by these enzymes, and that they need to be rebuilt for the attachment. So attachment will be delayed. So physical as well as physiological factors also affect the attachment. And because of this, the selective adhesion methods are limited success. So you are using selective adhesion. You are using the previous method, plating the cells. After half an hour, you take the supernatant out so that most of the fibroblasts are attached. Still, even if you are repeating this for a number of time, still there is a chance of fibroblast contamination, and you need to remember that the rate of proliferation of fibroblastic cell is far higher than epithelial cells. So after a few days or weeks, the number of fibroblasts will multiply. That will be overgrown above your fibroblasts. Then, what is the solution? You combine this selective attachment with other type of methods. For example, so selective detachment, we have already mentioned this. You can use enzymes such as dispase that will release epithelial cells before fibroblasts. So that is very important because the type of attachment of the cells to the surface or the intercellular attachment vary between fibroblasts and epithelial cells. So by using different types of enzymes, by using dispase, will detach these epithelial cells will detach before fibroblasts. So it is used this cannot be used in all conditions, in all types of isolation. For example, in the case of colonic cell, isolating cells from colon, you can use this dispase enzyme. And EDTA can be used to remove fibroblasts selectively from mixed cultures of keratinocytes. That is another method. So you are selecting using a different type of enzyme. Here you are using EDTA to remove the fibroblasts from the keratinocytes. And so firstly, principle of selective attachment. Then you can modify substrate to favor epithelial attachment. So depending upon the nature of the substrate, also the speed of attachment and the type of attachment vary. So you just change the substrate to which you are attaching. So for example, native or undenatured collagen used to select epidermal cells and breast epithelium. So you plate the surface with native or undenatured collagen. So that will further the attachment of epidermal cells and breast epithelium. So either collagen and laminin separately or combined, you can use it depending upon the type of cells. So this will form to encourage the expression of the differentiated phenotype in many epithelial cells. When you are differentiating epithelial cells, you can use this type of combination of the substrate modification. But this may limit their proliferative capacity because it is concentrating on differentiation. So the proliferation may suppress. So if you need more cells, this is not advisable. And there are different companies develop different types of flasks, culture flasks, with the different types of surfaces. For example, Primaria. It is a modified plastic from Becton lignin. It's a net positive charge. It is in favor of epithelial cells over fibroblasts. So this Primaria flask, its culture surface has a net positive charge and which favors the attachment of epithelial cells over fibroblasts. Another thing, another surface modification, you are using a feeder layer and over the feeder layer of cells you grow your epithelial cells. So first a monolayer of fibroblast. A layer of fibroblast is made on the culture surface. And you need to make sure that these fibroblasts are irradiated so that they will not multiply. Gamma irradiation is done and the multiplication ability of the cells are lost. So it will remain as a single layer and you grow epithelial cells over that. Because epithelial cells can grow over fibroblasts, but fibroblasts cannot. So this enhances the survival of many epithelial cells, including keratinocytes, cervical epithelium and breast epithelium. So feeder layer represents the growth of fibroblasts because of by the ability of epithelium to attach to the fibroblasts. As I said before, epithelial cells can attach to the fibroblasts like they are attached on the basal membrane. But normal fibroblasts are unable to do so. So the fibroblast, normal fibroblast will not grow in a flask where there is a feeder layer of fibroblast cells. And also the feeder cells, they are still growing, even though they cannot multiply. Their life cells, they release paracrine factors that enhance epithelial survival and block the action of TTF. So this is a modification in favor of growth of epithelial cells. This is one of the most favorable, successful method of enhancing epithelial growth and inhibiting fibroblast growth. This is a continuous inhibition, so that the paracrine factors or the feeder layers are there. So long as these two are there, the fibroblast will not attach to the surface. So this we have already seen physical methods such as density gradient. You are suspending the cell, depending upon the weight of the cell, the heavier cells will settle, that we have already seen. Then density gradient, you use different levels of, so in a tube you are taking media or particularly designed liquid with the different densities at different levels, so that the cells will reach to that to a density equal to their weight and they remain there. So density gradient separation. Then centrifugation can also be used. Then centrifugal illustration and flow cytometry, we will see the details later. So flow cytometry has a greatest resolution. You can get pure cells, pure cultures using flow cytometry, but its yield is low. And also technically complex and involve expensive equipments. You cannot adopt this technique in all the labs because you need a flow cytometer which costs nearly $1 crore or more. So in normal labs, standard labs, this technique cannot be applied because it needs expensive equipment. But it is very useful with the different types of cells. By adjusting the cell size, you can get single type of cells, very pure collection of cells. Then centrifugal elucidation. The principle is same as centrifugation or like a density gradient centrifugation. So this is the container where you load the suspension of cells with a medium. First, you allow a low density medium to pass through. Along with that, this structure will be rotating. Because of this centrifugal force, the cells get separated into different layers. So here we have a low density fluid. So the heavier cells will be settling down and the lesser dense, less heavy cells will be towards the end. As the fluid is flowing, so these cells will come out first. So you take those cells. Then what we do, after that you will allow a high density fluid to go through, so that the cell will not go down and you will get the heavier cells out. So you are changing the density of the fluid going through while the centrifugation is happening. So this will give a sort of pure collection of cells, but still there is chance of contamination. Then magnetic separation. This is one of the best methods for the separation of cells. So here you have an antibody which can attach to the cell surface receptors. This antibody is attached to an iron bead, so it is magnetic. Antibody which can attach to the cells which you are going to separate, and that antibody has a magnetic bead. So these cells and these antibody are mixed together. As a result, what will happen is the antibody attached to the receptors of the cell, and you pass it near to a magnet. What happens is this iron will get attached to the magnet and the cells will remain there. The cells without this antibody or magnetic beads will be moving away. So you get the cells separated. So you can either use this technique by positive or negative separation, because you can, so suppose this is a cell you wanted to separate, this is cells which you are going to use, so you will select antibody which can attach with these cells, so that is positive selection. But sometimes you may not get a particular antibody which can attach to the cells of your interest. Then what you do is, you take an antibody which can attach to the contaminating cells, so that cells will be removed and the remaining suspension will be a cell of your interest. So the positive selection is better than negative selection. But sometimes you may not be able to do that because of the lack of specific antibody which is specific to your cell type. Then, once this antibody is attached to the cell, you have to separate it. You put the cells through a glass cylinder, cell suspended in the medium. So the cells with the antibody attached and also cells without the antibodies. So you allow this to pass. So then there is a magnet on both sides or around the tube, and this is electromagnet. So when the cells are passing, electricity is passed through the magnet, and the magnetic property is there. As a result, the cells which have these antibodies with the iron particles attached will remain there, and the cells without the antibodies will go. So you remove the cells which you don't want. So once the movement is over, the flow is over, what you do is, you just switch off the electric current to this magnet. As a result, it will lose the magnetic property and the cells which are attached will come out. So it's quite easy method. You get a almost pure suspension of desired cell. So turning the current flow allow will remove the magnetism and the cells can be electrolyted. And after this, you can remove this antibody by using trypsin enzyme. So antibody, they are proteins, and if you put the cells with the antibody in trypsin solution, and the antibody will be digested and the beads are removed. But sometimes nowadays we can get these selecting antibodies which don't need to be removed. You can just culture it as such, so that the antibody will not be disturbing your culture. Then cloning. So as I said before, when you are using flow cytometry, you will get a less number of cells. So the only solution is, you will let them multiply and use them. And you make clones of the cells and use it. So you get a pure culture of minimal number of cells, and you grow them and let them proliferate until you get enough number of cells. So this is used when you can get really few number of cells. And you can also pool the clones. You can isolate different times, and later you can pool the clones and use. But the problem here is, sometimes the culture may senescence. They will stop proliferating. So you don't get enough number of cells. So then the solution is, you immortalize the cell. Cell immortalization we will learn later. You make the cell continue divide to make them divide continuously without senescence. So they will get immortalized like cancer cells by adding or including or incorporate transforming with the specific genes. You can immortalize the cells. And for the different types of epithelial cells, growing in different types of cells, use selective media. Then selective media can be employed to separate or to isolate desired cells. So several medias are there supporting different types of cells. Like a serum-free media, certain cells will not grow in serum-free media, and your cell of your choice can grow in serum-free media, and we use serum-free media. Then you can incorporate certain hormones or growth factors like hydrocortisone, isoproteinol, EGF. This will stimulate epithelial proliferation and It will not have any stimulatory effect on the fibroblast or other type of contaminating cells. So this property, and also you can eliminate TGF. You have seen this TGF and PDGF. They will suppress the growth of certain type of cells. So you add or you eliminate this from the media. So by modifying the media also you can have a selection of the desired type of cell. And here the advantage is this selective pressure is there in the medium continuously. So it keeps on suppressing the growth of unwanted cells. So it's a continuous process. Do not depend on one selective event. So in the previous methods you just select the cell, after that you are not putting any selective pressure or selective process. But here the selective process is always there whenever so long as you are using this media. So the other cells will not grow. And nowadays many of these media are available commercially. Then, so you have selected or you have separated the cells. And some of these methods depend upon specific surface receptors. Sometimes there can be different types of cells with same surface receptors. So they will get separated or selected. Or if you are separating the cells by difference of size, there can be different types of cells with the same size, density. So that can also cause contamination. Then, how will you select? How will you say that? Sometimes you can determine the type based upon the morphological properties. We can say, okay, this is that particular type of cell. Sometimes that is not possible. So you need more precise method for identifying the cells, whether the cells you have collected is the one you need or not. So as I said, morphology is one basic criteria. Most epithelial cell cultures have a characteristic tight pavement-like appearance. So they are tightly packed like a pavement, properly laid pavement. But sometimes, some type of cells, even if they are epithelial cells, they show greater plasticity in shape. So that shape may change. So you cannot separate or you cannot identify them based upon the shape. So some mesenchymal derived cells like 3T3 cells from the endothelium and mouse embryo fibroblast, these cells look like epithelial cells. So the shape cannot be adopted in these cases. Then what is the specific method? You are depending on surface markers. Each type of cell has got a specific type of marker on the surface. So reliable identification. You can depend on this identification based upon certain specific markers. As I said before, the intermediate filament proteins, they have been used for a long period of time as a marker, like cytokeratin group. They are found predominantly in epithelia. So if you can locate the presence of cytokeratins, so that will ensure that the cell culture you have is that of epithelium. The cytokeratins are a large group with a considerable diversity. There are different types of cytokeratins, and different types of epithelial cells have different types of cytokeratins. So that can be used as a marker for identifying different types of epithelial cells. Not just identifying epithelial cells from other cells, and also this same marker, because there are different type of cytokeratin, the same marker, the cytokeratin marker can be used to identify in between epithelial cells. Different antibodies are available for different cytokeratins. So as a result, they are used to identify different types of epithelial cells also. This is an example. This is one type of anti-cytokeratin, anti-AQP2, anti-cytokeratin 8 to 18. So they all give different types of mark. So you can identify different using different types of antibodies. You can identify different types of epithelial cells and also the different stages of the same type of epithelial cells. Desmosome junctions, that's another identification. They are specific to epithelia and this can be detected by electron microscopy. So you don't need any antibody here. You can detect by electron microscopy. So like protein like desmosomal proteins like desmoplakin can be determined by immunostaining, like using antibody. Specific antibody, you can stain and determine whether this protein is there or not. Then, as I said before, differentiating cells, same epithelial cell, depending upon the stage of differentiation, the cytokeratin may vary. The junctional complexes of the desmosomes, tight junction seen characteristic association depending upon the stage of differentiation. The association between desmosomes and tight junction in specific way, so that will make you identify the differentiation stage of the epithelial cells. In case of certain epithelial cells, when they become confluent, they generate a so-called dome. As I said before, the cells grow in a layer. When they are confluent, this type of cell, what they do is, they detach from the surface and they form a dome. So the middle part is detached from the culture surface and the sides are still attached. So it forms a dome. So that will give you some idea about the type of epithelial cells. Surface antigens. A number of specific surface antigens are there. They are specific to specific type of epithelial cells. Like transmembrane mucin, like glycoproteins. Include epithelial membrane antigen, EMA, human milk fat globule, HMFG1 and HMFG2. They can also be used for identifying epithelial cells. So these antigens are strongly expressed in differentiated ductal epithelial cells. So that is a clear identification of differentiated ductal epithelial cell. That is a human milk fat globule HMFG1 and HMFG2. And they are present in varying degrees in different type of epithelial cells. That is another way of identification, whether these antigens are there or what degree they are present. And in addition to, there are so many specific markers are there. They are all available commercially nowadays, so you can easily identify the cells. So different methods of separation, isolation, identification we have seen. And the nature of epithelial cells make them a suitable in vitro model for research, for carcinoma, candy cancer studies, differentiation studies, so many different types of drug testing, etc. So the different nature, that is their unique shape and attachment properties. So some cells are detached very fast, so handling them is very difficult. But in the case of epithelial cells, they can attach firmly to the surface. So that can be used, especially when you are staining the cells using different steps. You want a cell which remains firmly attached to the surface. So because of that property, so unique nature of attachment, like epithelial cells are preferred choice. And there are specific markers for identification. You can easily identify what type of epithelial cells, whether it is an epithelial cell or what type of epithelial cell at what stage of differentiation, etc. So that also make it a preferred choice. And there are different modified forms. You have seen different types of naturally available cancer cells, breast cancer cells, skin cancer cells. So the different types of epithelial cells, different modified cells, and seen in different parts of the body, ductal epithelial cells, in the kidney tubules, in the digestive system, in the lungs. So many different types of cells are there. That also makes them a preferred choice and easy to maintain. It's not very difficult to culture them. Just seed them, give the media needed, and they will grow happily. So that is a good thing about the epithelial cells. And reasonable doubling time. You can get enough number of cells within a decided or within a short period of time. Not as fast as fibroblasts, but it has got a reasonable doubling time or 24 hours or something like that. So you can get enough number of cells within a short period of time, and as I said, good attachment. So they get attached to different types of surfaces. You don't need a specifically modified surface for attachment. Normal, general cell culture flasks or plates are enough for attaching the cells. So that also makes them a suitable choice. So we isolate the cell using mild enzymes like pronase, etc. Or you can even use collagenase, dispace, and also even certain cases you can use trypsin. Then you can separate cells using physical methods like density gradient separation or gravity assisted separation, or you can use magnetic beads for their separation or differential attachment. And once they are separated, you can identify them using different types of markers, surface markers and cytoskeletal proteins and desmosomes. Different types of antibodies are available, and you can separate them also by flow cytometry, and different types of media can be used to favor the growth of epithelial cells and suppress the growth of other contaminating cells. And because of these properties, they are preferred choice of cells for research. Thank you.